thanks very much for coming. It's a great honor to get the invite to come to speak, and what I want to try and do today is talk about some topics that I certainly have not talked about before at the RCI. I was digging back into the past history of my talks here, and the first talk I gave was on invasive species, which is an interesting topic that I've worked on for some time. And then the last time I spoke was at the beginning, or about 14 years ago, when I talked on genetically modified organisms. But they're not my actual research areas in any main way. What I actually spend most of my time thinking about and worrying about and waking up in the middle of the night in a cold sweat about is plant sex. And so that's the topic, and it may not be something that you wake up in the middle of the night in a cold sweat, but I hope by the end of this lecture you'll have a better appreciation for the really interesting aspects of the sex lives of plants. So that's my goal. There's a sub-theme here as well, and that is that although modern science has become awfully complicated, it is still possible to make discoveries simply through natural history observations and understanding the broad issues of a given field. And so what I hope to show you in the talk, which will go from natural history right through to genomics, is that now modern evolutionary biology has taken on board a whole bunch of different approaches, but the starting point is just having good eyes, being out in the field, and looking for research problems, a truly Darwinian approach. So that's the kind of one of the main themes. Okay, we got our lights down here? Yeah, okay. I want to sort of start with this because I always like to start, I always feel that biology needs to drive things. You know, often you start off with Darwin, 1887, or Fisher, 1930, but let's start with biology here. And this is a photograph I took several years ago in South Africa in a national park. And what's remarkable here is that there's about 60 or 70 different species of flowering plant, and they're all flowering at the same time. Most of them are animal pollinated, but they're all using different strategies to gain reproductive success. And so for somebody like me, this presents a puzzle. Why are there so many different ways of bringing about the fundamentally important aspect of evolution, which is transmitting genes from one generation to the next through mating? Why don't plants all do it the same way? Well, they don't, and so I'm very interested in all of the different strategies that have evolved that promote mating success. I want to start by saying that a lot of what I'm going to discuss is actually ideas and experiments and work that is done in my laboratory by a great group of graduate students and postdocs. So this is very much a collective effort, and this is the group as of 2015. And I'm fortunate to have people not only from Canada, but from various parts of the world, and that's great because we get to learn about new systems in different countries. On the right here is basically the structure of the talk. I'll start off, with, and I don't want to be condescending here, but let's call it Plant Sex 101. We need to get some ground rules so that everybody appreciates that plant sex is different from human sex. Okay? Then we'll move on to how do you study plant sex? I mean, how does somebody like me go about posing questions and doing research? And so we'll spend a little time briefly on the idea of the approaches. I want to pay great respect and homage, if you like, to Charles Darwin, because I'm sure you all appreciate that he's a serious hero for those of us in evolutionary biology. And obviously The Origin was the most important book ever written in biology, and maybe it could be argued in science. But I don't want us to lose sight of the fact that Darwin was not only a great evolutionary biologist, he was a fantastic botanist. And he wrote 6 books on plant biology and spent more of his life working on plants than any other group of organisms. So any zoologists out there, bear that in mind. He was a botanist first. And so I'm going to talk a little bit about some of Darwin's work and how that established essentially a foundation for much of what I'm going to talk about today. We'll then get into the form and function of flowers. I'm going to talk about couple of problems that I've spent quite a bit of time working on in my career. One is the evolution of self-fertilization, because it presents a bit of a puzzle. And then we'll also look at the evolution of separate sexes, why some plants have given up hermaphroditism for being males and females, the sexual system we have. And I'll sort of point out that that does present a puzzle that goes right back to Darwin. Then I'm going to sort of finish off with some fun and games, plant sexotica. We're going to look at some really weird sexual systems that I've worked on, some of which we understand a little bit about, and some where there are still some really big unresolved issues. And then I'll finish with a few take-home messages. Well, I think it's fair to say that botanists are fairly obsessed with flowers. I'm sort of a bit reluctant to push that strongly because my colleague, Professor Nancy Dengler, is in the audience, and Nancy has spent most of her career working on leaves, okay? But there are many of us who are obsessed with flowers. This is just a picture at the International Botanical Congress. And so the question is, you know, why the fascination? Well, I mean, there are reasonably compelling arguments for why flowers are important. They're the sexual organs of the flowering plants. And of course, if we really think about it seriously, if 12,000 years ago we hadn't invented agriculture, and if plant breeding hadn't developed to the extent that it has today, we would not have the sort of civilization that we have today. In other words, an understanding of plant sex is integral to plant breeding, to biotechnology, to genetically modified organisms, and in fact a lot of other topics as well, such as invasive species, and in fact why some species are extraordinarily rare. They're having problems reproducing and regenerating. So, you know, it really brings a lot of topics together, and I like to work on it because it also provides an opportunity to study plant-animal interactions. So one can also be a zoologist at the same time, because many plants are, of course, animal-pollinated. But the real reason I study plant reproduction is because at heart I'm an evolutionary biologist. That's what I teach at U of T, and I use plants as examples of adaptation and natural selection. And as Darwin first pointed out, plants are wonderful organisms for the study of adaptation and evolution. So there are good reasons why one might be interested in flowers. So let's jump right into it, and this is the sort of 101 part. Let's just step back and ask the question, what are the distinctive features of plants that influence the way they have sex, the way they reproduce? Well, first of all, obviously, one of the most important things is that they're immobile. They're sedentary. They don't move around the landscape as we do. And that presents a problem for mating. It means that in order for a male or the gametes from the male side of a plant to be transferred to the female side of the plant, there needs to be agents for that dispersal of pollen. And so that defines a whole field of plant reproductive biology, and it's called pollination biology. It's a very exciting field where people study the various agents that move gametes, male gametes, from one plant to another. That arises, of course, because of the immobility of plants. Immobility also is associated in the whole of the kingdom of life with hermaphroditism. It turns out it's not just a plant trait, but it's highly correlated in animals as well with hermaphroditism. So if we look at, you know, what animals actually are hermaphrodite, the majority of animals have separate sexes like us, but those that are hermaphrodite are very often immobile, things like corals and other animals that are not particularly vagile. So hermaphroditism may be sort of viewed in some ways as a bit of a safeguard, particularly under low density conditions where there aren't mates for cross-fertilization. Another aspect of plants that I think is very important to consider is that many have dual reproductive strategies. That is, that they don't just rely on sex, they often can clone as well. And a vast number of flowering plants, particularly those that are perennial, have the ability to propagate clonally. And so now we're considering two forms of reproduction, both sexual and asexual. And so some of the things I'm going to talk about are how those different reproductive modes interact. Another aspect of plants that's important is that unlike us, where we have discrete gonads, reproductive parts, and reproduction is fairly easy to understand, plants produce multiple reproductive structures, hundreds of flowers. A huge tree in a tropical rainforest each flower may be pollinated by pollen from a different individual. So we can have multiple paternity. It's a little strange for us to think about, but plants can simultaneously mate with many mating partners. And of course, that increases the genetic variation of the seed crop each generation if many males are represented among that seed crop. So very complicated mating, and that all comes down to the modularity of plants. Plants are like Lego. They're made of modules. And so we have shoots and we have inflorescences, and plants can get vast in size with different parts of the plant experiencing different ecological environments, particularly where cloning is very large. And then also, just to finish off, plants have enormous life history diversity. When you consider that the shortest lived plant may in fact go from germination to mature seed within a month, so that's a very fast growing annual. But we also have things that can grow for thousands of years. I mean, you know, the record is the bristlecone pine in the White Mountains of Western North America that can live for 4,000 to 5,000 years. So we have enormous longevity, and it's no coincidence that there is a non-random distribution of the kinds of sexual systems with longevity. Short-lived things like annuals are often highly self-fertilizing. Plants that live for long periods of time are generally cross-fertilizing. So here's a very simple scheme that allows us to just think about this complexity of plant reproduction. We can start off using the term reproductive system, and we can say, well, the two fundamental types are sexual reproduction, where gametes fuse to form a new seed, or asexual reproduction, where an individual just grows, and as it grows, it makes copies of itself. No genetic change there. It's just the photocopying of the genetic material through growth. Then if we just consider the sexual cycle, we can look at see whether the plant is a hermaphrodite, that is a maternal and a paternal parent at the same time, and the vast majority of plants are hermaphroditic. But we can have separate sexes, such as we have, and most vertebrates have separate sexes, and the two sexes are called male and female. But that's actually not a particularly common condition in flowering plants, and I'll return to that a little later on. And then if we just consider those hermaphrodites, they can be fully cross-fertilizing, in which every new seed is a combination of genes that come from a maternal and a paternal parent. That is the products then of crossing between plants. Or some of the seeds may be self-fertilizing. In other words, that plant is mating with itself. And it turns out that's a very common way of mating in many of our crop plants. Many of our cereals are fully self-fertilizing because breeders like to produce very pure and uniform crops, and selfing can give you that. Okay, so let's just be clear on this. Let's take just a flower. This is a lilium from California. And what we have there is the showy petals, which attract pollinators. But if we look at the bits a bit more carefully, part of the flower is male and part is female. Now, of course, for Darwin was very interested in this because he was obsessed with the issue of inbreeding. And there's some interesting issues there. Why was that? It was partly to do with his family and the fact that his children had quite bad ill health. And he worried that, in fact, his children might be in part inbred. I won't go into the details of that, but he was very, very interested in inbreeding. And he wanted to study it, and so for him, the way to study this was to study plants, because here we have hermaphroditism, and there are opportunities to do experiments to see whether, in fact, the products of self-fertilization are generally less fit, their performance is not as good, as those individuals that come from cross-fertilization. And, of course, plants are very easy to cross. Some of the major discoveries in the history of science come from working with plants. Mendel's laws were not developed from working on animals. They came from working in crosses between pea plants. So plants are wonderfully tractable organisms for doing genetics and for doing crosses. So most plants are hermaphroditic, And of course, what he was interested in, what Darwin was interested in, is just the issue of, well, if the sex organs are so close, here's the anther, here's the stigma, how does this plant stop itself from mating with itself, knowing full well that that's going to result in less fit progeny? Well, it turns out that this particular plant has a complex mechanism of self-rejection. It can actually recognize its own pollen, and when that pollen tube is growing down the female sex organ, the style here, it's rejected by the maternal tissue. And the genes that control that have now been cloned. We understand that 50% of flowering plants have a complicated physiological self-recognition system. That's why when you go to the nursery for some plants, they'll say, like apples, buy 2. You need 2 plants. They have to cross-fertilize in order to get fruit. Many of our plants, our provincial flower, trillium, is a self-incompatible plant. In order to get seed, crosses between individuals are required. So that's hermaphroditism, and it's very common. What is much more rare is this plant called Early Nancy, named after Nancy Dengler, who's in the back there. This is an Australian geophyte, and we worked on this plant for some time in the 90s. I had a PhD student, Andrea Case, who spent her whole thesis, or a lot of it, in Australia. And so here what we've got is 2 individuals, one individual here that is large with showy flowers, and this individual here with smaller flowers. If I tell you that one is a male and one is a female, see if you can guess which one is which. Okay, I'm going to ask you now, so who thinks this is the female? Okay, that looks like a majority. So that means that who thinks this is a female? We've got a few in there. Okay, let's see who's right. Now, the reason that most of you got it wrong is because you were thinking, probably not thinking sensibly, and you were forgetting about a peacock and a peahen, or a male cardinal and a female cardinal. I mean, a general rule when there is sexual dimorphism, which is what we have here, we have 2 sexes, and they look different, is that males tend to be big and showy, and females tend to be small and drab. And that was explained by Charles Darwin as the product of sexual selection. Males compete with one another for pollinators, which are then transferred to females. The constraint on fitness in female is not whether she's going to get pollinated. She'll get pollinated. It's whether she's got the resources to mature those seed and fruit. And that is the major constraint on passing genes into the next generation for a female, her condition. So allocating to big flowers is a waste of time. 1 bee visit will do the job. Whereas for a male, having bees constantly coming and transporting your gametes to as many females as possible is the name of the game with regards to evolutionary fitness. So this is simple sexual dimorphism. However, this kind of condition is quite rare in the flowering plants. It only occurs in about 7% of species. You know full well some of them, papaya, kiwi fruit, marijuana. They're all examples of plants that have separate sexes. And that condition, by the way, has evolved independently about 100 times. So it evolves quite frequently, but it's relatively rare in terms of species numbers. Now, I mentioned cloning. Here is the classic cloning plant. This is the plant I did my PhD thesis on. And this plant has an amazing capacity for clonal propagation. One plant can form a hectare of plants in a few months simply by growing rapidly and cloning. It's called water hyacinth. And it, like many perennials, has a dual reproductive system. It can clone, make copies of itself through growth, and then it can also flower. And usually the conditions under which it clones versus it has sex are different. And it's nicely illustrated here with this peripheral flowering pattern in this one big clone here. There's no flowering here. These plants here are flowering because they're touching the bottom. Their roots are in the soil. And that is a cue that goes back to the home of this plant. It's from the Amazon. And it often gets itself into shallow lakes where it clones like mad during the wet season when the lake is the depth of water is high. But during the dry season, and there is indeed a dry season even in the Amazon, the lake's going to desiccate. It's going to dry out completely. And so the only way that plant can persist during the dry season is through seeds. So as soon as those roots hit the soil, they're cued to start flowering and make seed. So we have these different ecologies, if you like, for the different kinds of reproductive strategies. Okay, so I mean, what do we need fancy equipment for this? Well, no, actually, a lot of what I do is pretty straightforward. I just decide at some point in the year, I'd like to go to Madagascar, or I'd like to go here. Turns out Thursday I'm going to Australia and New Zealand for 5 weeks. Travel is what it's all about. It's one of the great things to do in life. And if you can find a good problem, then you can go and chase it down and do some research on it. And when you do that, what do you need? Well, you need some seed envelopes, a pair of binoculars. You know, why go into these anaconda-ridden marshes in the Pantanal when you can actually scan and see if your plant is there from the boat? You need a pair of bluntstones. They're great boots to go into, you know, to do field work. You need a hat. Skin cancer is a big issue now. Camera and a vehicle. It's dead easy. That's called natural history. And there's nothing fancy about it. All you need to do is get a grant and have a compelling hypothesis. Okay, so, you know, we can take that kind of classically Darwinian approach to studying plant sex, and a lot of what I do is simply this. There's nothing fancy about it. It's just a question of going out into the field, sampling, and sometimes doing field experiments. But you don't have to do field experiments in faraway places. In fact, I've done quite a lot of field experiments in my backyard in Etobicoke. And, you know, we published some reasonable papers that have come out of that. And this is just a question then of growing plants in a greenhouse, having a good question, and doing experiments. This is a classical Darwinian approach. As you probably know, when Darwin came back from the Beagle, gone around the world for 4 or 5 years, he came back, he wasn't very well, he got married, and he decided he was going to stay put. He stayed at Down House for the whole rest of his career. He never left England after that. But he had a greenhouse, he was relatively well off, he had a horticulturalist gardener, and he just did a ton of experiments on plants. And he found some really, really interesting results. So, you know, one can do these kinds of experiments if you have a decent question. This was an experiment we were doing on a member of the potato family, Solanum, where we were looking at pollinators and their visitation patterns. So that's kind of the low-tech side of how you study plant sex. But we're experiencing a revolution now in evolutionary biology. And one of the greatest shocks to me, because I would have never predicted this 15 years ago, is that now we're doing genomics. Now this amazing technique of sequencing DNA allows us to do all kinds of things that we weren't able to do 10 or 15 years ago. So many areas of biology have been revolutionized now by this thing, the DNA sequencer. And the DNA sequencer allows you to look at DNA sequences between individuals, between populations. We measure variation at what is called SNPs. So what is a SNP? Well, it's a single nucleotide polymorphism. It's where a substitution has occurred through mutation in the DNA strand, and that's a great way of measuring variability. We all differ from one another in thousands of single nucleotide polymorphisms. And then we can look at the consequences of different mating patterns on genetic diversity right across the genome, and we can measure the relationship between populations using these DNA sequences. We can even do, as I'm going to mention in a minute, we can actually get and look at, well, how long has this species been in an area? We can use coalescence models. These are models where we take the DNA sequences, and under some assumptions, we can figure out, knowing mutation rates, how long species have been in a given area and when speciation occurred. So it's an amazingly powerful technique. But let's go back to Darwin, and let's talk a little bit about his books. These are the 3 most important books in plant reproductive biology. They essentially started a whole series of subfields. This is a wonderful book on orchids, written in 1862. There's a thing among evolutionary biologists, you may have heard of sort of ideas about the adaptationist program. And there's the idea that adaptation became a centerpiece of 20th century research in evolutionary biology. But actually, if you trace the beginnings of the adaptationist program, it goes back to Charles Darwin. This is a book about adaptation. He was studying the structure and function of orchid flowers and interpreting them in terms of mechanisms that were promoting cross-fertilization. So it's a wonderful book. This also is a remarkable book. I often have to defend Darwin to people who often say things like, well, wasn't he just a product of his time? What about Wallace? What about Lamarck? Those other scientists were great scientists. But Lamarck didn't do any experiments. He just wrote down ideas. And Wallace certainly was brilliant, but he wasn't in the same league as Darwin. Darwin spent years doing experiments. And let me give you one example. He was a true experimentalist. This is a book with a rather quaint title, The Effects of Cross and Self-Fertilization on Members of the Vegetable Kingdom. I love that, vegetable kingdom. Okay, not just plants, you know. In fact, it wasn't members of the vegetable kingdom because, I mean, not vegetables per se. He looked at 52 species in 34 families and conducted controlled crosses and selfs. In other words, he self-pollinated the flowers with a brush or forceps, and then he cross-fertilized them, and then he got the seed, and then he compared the growth. And in almost every case, he found that the progeny that arose from cross-fertilization were way better at performing in terms of their height, the number of flowers. They were fitter. So cross-fertilization gives you more fit progeny. So, you know, that's a pretty big research program, 50-odd species. But it doesn't stop there. For those of you who know morning glory, one of the species that he worked on, the morning glory, he did this every generation for 10 generations. It took him 10 years. He had one generation each year. And he selfed and he crossed. And then he grew the progeny out, selfed, crossed. 10 years. And then he observed what happened to the progeny after 10 years of inbreeding. And he saw that gradually it declined. And this then led him to conclude that nature abhors self-fertilization. In other words, inbreeding isn't a good thing. Now, we know why now, because the genetic basis of inbreeding depression is well known. It's the deleterious genes that we all carry that are normally in the recessive condition. Every one of you in this room are walking around with probably 8 to 10, 14 lethal genes that would kill you if you were homozygous for that lethal, but you're not. Fortunately for you, the other parent had a dominant allele at that gene locus. So inbreeding results in homozygosity for deleterious genes. Darwin didn't know the genetic basis of inbreeding depression, but he knew that it obviously was an important problem, and that's why he turned to this book, his most important book in my view, which was to try and interpret the wide array of different kinds of strategies that plants have evolved that limit the harmful consequences of inbreeding. And I want to tell you about a few stories here that I've become involved in over my career that essentially look at some of Darwin's ideas. And we're going to start with my favorite mechanism, and it's called heterostyly. So I'll explain what that is. But independently, in 28 different families, a particular form, a particular kind of flower type or types has evolved which limits opportunities for inbreeding. So it occurs in all of these families. Some of these families may be unfamiliar to you. You certainly should know the iris family. Purple loosestrife is in this family, which is our wetland invader, and many of you may know primula. This is, by the way, the plant that makes cocaine in the Erythroxylaceae. But in fact, if you're wondering about whether or not you can see and look at heterostylous plants, I would say absolutely. They're really common here in the spring. These are photographs from my garden in Etobicoke. I happen to have a lot of heterostylous plants because I like them, but they're common species. Forsythia is heterostylous. This is a primula. This is bog bean that grows in a bog that I have in my garden. And also here's purple loosestrife, one of our serious invaders. These are all heterostylous plants. So you can see these, and when you do see them, I hope you remember this lecture because it would be interesting to go and have a look at them carefully, look at the naughty bits, the sex bits, because what you're going to find is it's quite interesting. There's quite a lot of interesting variation occurring. And let me just briefly explain what heterostyly is. There are two forms. You can either have two types of plants, one with a long female structure. This is called the style, and that's the stigma, which receives pollen. These are the anthers, and these are called stamens. So this is the long-styled form, this is the short-styled form, and that's what you get in that system. It's evolved independently in 28 families, and the only seed that you can get is crosses where these arrows are. This pollen is transferred to here, this pollen is transferred to here. How is it transferred? Usually by bees. These are all heterostylous species, are all animal pollinated, and the pollinators pick up pollen on different parts of their insect body and hence are transferred to the stigmas that the pollen works on, or in other words, we say is compatible with. So this is a mechanism to enforce crossing between plants. And in fact, if you go into natural populations of these plants, you usually find a ratio of 1 to 1. It's as if we have two sexes, just as we have here. The equilibrium, the evolutionary equilibrium, is the same as the sex ratio. It should be 1 to 1. And it arises by what we call disassortative mating, mating between unlike phenotypes. Now, tristyly is more complicated because there are 3 forms, but it's the same principle. The only difference here is that flowers have 2 stamen levels, and therefore we have the remarkable situation where the plants are producing 2 kinds of pollen with different fates. This pollen, for example, when transferred to this stigma, will give you seed. If you take this pollen from the same flower and put it on here, you'll get no seed. There's nothing like that in the animal kingdom. It's as if in this room, males were producing 2 kinds of sperm, one sperm that works on a set of females and another set of sperm that works on another set of females. There isn't such a thing in the animal kingdom. This is a complicated system, okay? And Darwin was fascinated with this, and how did it work? He in fact said about this particular condition, it is the most complex marriage arrangement that I have ever observed. 3 intercompatible hermaphrodites co-occurring within a single population. Now, we know quite a bit about the genetics. This is controlled by a single locus with 2 alleles. This is just the genotypes here, so a dominant S allele causes the plant to be short. The genetics of this one is a bit more complicated with two gene loci. So I was very interested in this system as a graduate student at Berkeley, and when I came to Toronto in 1977, I was very determined to actually see if I could sort of test Darwin's hypothesis on the adaptive significance of heterostyly. And this arose because in this book, he makes this wonderful statement from my perspective. He says, I do not think anything in my scientific life has given me so much satisfaction as making out the meaning of the structure of heterostylous flowers. In other words, what he's saying here is, I have an adaptive hypothesis that explains this and why it's evolved. That's essentially what he's saying. He's suggesting he knows how this functions in natural populations. So anyway, this is an old story, but in the sort of 70s and 80s, my lab started to investigate this, and we thought, well, can we find a local species that we can test Darwin's hypothesis? You know, it has to be heterostylous. And there was a wonderful species to work on, and that's some that those of you who go up to cottage country will know. It's the pickerel weed, which is all through the Muskokas. It's a very attractive plant. It's tristylous, and so we set about testing Darwin's hypothesis that this is a mechanism to promote cross-pollination, but in a very specific way. I mean, just going back to that diagram, we are predicting this, that pollinations are largely from long level anthers to long stigmas, from mid-level anthers to mid, and short level anthers to this. Is that actually what one finds? Okay, so I was lucky. I had some really great graduate students at the time, and so this was the pickerel weed team. So Stephen Price was one of the first people. He's probably some of you know him from Toronto. He was with World Wildlife Fund for many years. And so Steve did the first experimental test, but we did later ones, Deborah Glover and Lorne Wolfe. Lorne was particularly interested in looking at whether there were 3 layers of pollen on the bee's body. Because this is the way it works. As the bee enters the flower and probes for nectar, pollen is deposited at 3 different positions in any one flower. There's only 2 pollen types, so it will be 2 levels. But at the population level, those bees should have 3 layers of pollen on their body. So being more formal, we can say the null hypothesis would be that if we sample pollen on a bee's body, it's not randomly distributed. So basically, without going into all of the details, we tested Darwin's hypothesis, and we were able to do this because pickerelweed has trimorphic pollen. The pollen from those 3 anther levels differs in size. Here are the 3 pollen types. This is the large pollen from the long level anther, the mid-sized pollen, and the short level pollen. So if we look then on a stigma at the end of the day of pollinating by bees, we should be able to reject the hypothesis of random pollination. In other words, if we know how much pollen is produced in the population, we simply say, is there this preferential transfer to each of the 3 stigmas? And we found, in fact, that was the case. There is good evidence in support of the Darwin hypothesis. So Darwin was right. You know, in a way, Darwin could have done this experiment. I mean, okay, you know, he may not have had a fancy SEM, but you don't need that. You can actually use a light microscope, even a binocular scope. So it would have been possible for him to test this hypothesis in the UK, where there are many heterostylous plants. And there are many cases like that, where Darwin had a great idea, but he had so many other things going on that he didn't actually test his hypotheses. And that's why people like me like to read Darwin very carefully, because we can often find these neat hypotheses that can be tested. Okay, I'm going to switch gears now and say that I became obsessed with this family, the Pontederiaceae. It's a group of aquatic plants. And during my PhD thesis at Berkeley, I was aware of a big controversy that involved Charles Darwin, but also J.B.S. Haldane, the famous population geneticist. And it was regards to water hyacinth. I've already shown you an image of water hyacinth. That's that highly clonal species that we saw in the canal in Florida. Darwin was sent material from southern Brazil by the Muller brothers, these 2 fantastic German naturalists who got fed up with Germany and moved to southern Brazil and spent the rest of their lives publishing papers in Nature. As far as I can see, they published whole loads of papers in Nature during the period 1860 to 1888. And some of the material that they sent to Darwin, because Darwin was a great correspondent, and he loved the Muller brothers because, you know, Darwin would say, could you go out and look for this? And they'd send him pressed specimens and seeds, and he would grow them in his greenhouse. One of the plants the Muller brothers sent him was water hyacinth, and he deduced, based on the position of the sex organs, that this plant must be tristylous, because he was sent only the long and the mid-morph. But these 2 morphs, if you recall, going back to that image, those 2 forms, okay, so he's got this form, the long form, and the mid form, but these have short level anthers. So there should be a short form. But among the collections that the Mullers sent, there was no short-styled form. So Darwin wrote in that book, The Forms of Flowers, he wrote there should be a short-styled form. It was called Darwin's missing form. And then subsequently many scientists tried to find it. This is just one example. After J.B.S. Haldane, who was a very strong communist, got fed up with Britain and went to India. He was very unpopular with the British government because he used to hold these amazing rallies in London and get people fire and brimstone against the elite. And he goes off to India, and one of the first things he does in India is he tries to find the missing short-styled form. And he has, we talk about public outreach, but this must have been one of the largest public outreach studies in the world, and it was done actually in the 50s and 60s. And he had Indian school children out throughout India looking for the missing short-styled form. Where found? And this is a paper that he published in 1960, not long before he died, in the Journal of the Bombay Natural History Society. In fact, by the time I got to Berkeley, it was generally assumed that the species had gone extinct, or the morph, the short-styled morph, had actually gone extinct, and that there were really only two forms there. And then often what happens is, I went off to the Amazon to do a thesis on rice weeds, and within a week, I'd stumbled on the missing short-styled form. I mean, I didn't go to the Amazon to look for it, and in fact, I defended my thesis project to my committee, and it was something completely different. But I wrote to my supervisor, Herbert Baker, and said, I think I've discovered Darwin's missing short-styled form. Should I continue with the work on rice weeds? And I got this nice handwritten letter back saying, drop the rice weeds. And I've been working on water hyacinth ever since, so it's a bit of an obsession. So I was very lucky, but I should say there was no plan here at all. Okay, so just to say that often one continues with these projects, so this was an early distribution. It turns out the short-styled form is restricted to lowland Brazil and South America, and this is the introduced range. This is the introduced range in Florida, and it's all through India and Australia and so on, but the short form wasn't there. Okay, and the reason it's not there is because the long form and the mid form cannot produce short, because the gene for short is restricted to that morph. It's a quirk of the genetics. So we have what is called a founder effect. This goes Ernst Mayr's idea that only a small portion of the genetic variability moves from one part of the world to another, and the short simply was not represented. So we did that work many years ago, and I'm pleased to say that some recent collaborations with some colleagues in China, Yuanyuan Zhang and Dayong Zhang at Beijing Normal, we've now just published a full global survey to test the hypothesis of a founder event and a restricted bottleneck using a whole series of genetic markers. And what we found is amazing reduction in diversity, as is predicted from the founder event, in the introduced range. In fact, we had populations from many, many places throughout the world, but 80% of these alien populations were genetically uniform, just completely all one genotype. 75% of those contained the same genotype. So there was almost no diversity compared with the native range. So that's sort of consistent with the earlier finding of the restriction of the short to South America. And I'll just say that I'm continuing my work on water hyacinth. This was a photograph taken in April of this year. We've just recently got a large grant to work in Brazil for the next 3 years. And I should say the funding is not through Canadian funding agency, it's through Brazil. And I'm collaborating with Nicolai Cunha here. And what we're doing is working in the Pantanal, which is the largest wetland in South America, where the water hyacinth is native. And what we're doing now is using DNA sequences to look at the levels of diversity actually in the Pantanal, not only in water hyacinth, but in its sister taxon, this species here, which is particularly interesting because it's very closely related, but it's not at all aggressive. So we've got this really serious invader, and this species a non-invader, and we're interested in questions to do with what makes one species so aggressive and the other one less aggressive. Now, continuing with this group of water plants, let me just tell you a little bit about some work we've done on selfing now, because up to now I've mostly been talking about mechanisms promoting outcrossing. Let me talk to you about the work on selfing, and this goes right back to my major professor, who I've alluded to, Herbert Baker, who was at the University of California at Berkeley for many, many years and has a law named after him. It's a very simple idea, but what Herbert said was that colonization, particularly long-distance colonization of islands or any dispersal event in plants, will favor individuals that can self-fertilize, that can mate with themselves. In other words, if you are self-incompatible, like the lilies, where you need a partner, then you have to have the partner dispersed as well. But the problem is many plants get dispersed as single seeds, particularly in aquatics, because they get on the feet of waders, and waders have these long migration routes. And therefore, if we get a single individual, that germinates, it's pretty much forced to mate with itself. There aren't any other mates. That will act as what we call a selective sieve that favors plants that can self. So that's called Baker's law, and it predicts that islands should have more selfers on them, plants that can self-fertilize, than mainlands, the mainland areas from which the island flora descended. And so we did a specific test of this, looking at the annual water hyacinth, Iconia paniculata, which was particularly useful for our test because it's very common in the north of Brazil, but occurs on the islands of Cuba and Jamaica and Nicaragua and also Mexico in Central America. And what we found was entirely consistent with Baker's law. Within this species, so we're talking about one species here, we have outcrossing forms with big showy flowers in Brazil, and then on the islands here, we have these little small flowered selfing forms. And when we've looked at DNA sequence variability, we've learned two things. One, we can say unequivocally that these populations at the northern part of the range are descended ultimately from Brazil. So by long-distance dispersal over long periods of time, seeds have been inadvertently carried by waterfowl, most likely, and established, and that establishment, through Baker's law, has favored selfing individuals. Mutants that do occur at low frequency in Brazil, but they're the ones that get established. And it turns out that shift from outcrossing to selfing is not a particularly complicated genetic change. We've also looked at the genetic architecture of this by crossing the plants, because they're the same species, and it turns out that we've discovered multiple independent shifts from outcrossing to selfing. So this is not one evolutionary event. It's replicated in evolutionary time with multiple independent transitions. And that moreover, these selfing variants that have established, that selfing is governed by a set of genes, recessive genes, and we find different recessives responsible for this transition. It's very interesting. So essentially what we're getting is the same functional phenotype, selfing, but through different genetic pathways depending on the particular origin. The other thing that we did, so we have evidence for Baker's law. We can show that it's a pretty simple genetic change. The one thing that I was always, and this is where DNA sequences become so fantastic, one of the problems I always had with this is people would often ask, well, how do you know humans didn't introduce it to the islands? This is a weedy plant, particularly in Cuba and Jamaica, where it infests rice fields. And so there was always this sort of nagging worry in the back of my mind that, well, maybe it wasn't birds. You know, maybe they've only been there for a short period of time and they came in as a rice seed contaminant. And that happens a lot with rice. You know, seed is imported from one part of the world, and there's always these tiny seeds that then become weeds. Well, that's where we can use DNA sequences. And my student Rob Ness was able to do a very nice coalescent analysis of DNA sequences that estimated that the long-distance dispersal events to the Caribbean were 125,000 years before present. Well, remember, agriculture's only been around for 12 to 14, so this goes way back, even though we accept that some of these estimates have got fairly large confidence intervals. It certainly happened before agriculture was around. And just to sort of finish off on the genomics, and this may be overly complicated, I'll just simply say for those of you who've been following our abilities to do wonderful things with the genome, I have a new PhD, or have a PhD student right now, Ramesh Arunkumar, who's been now looking at the genome of these selfing populations in Jamaica and Cuba, and has actually looked, well, to test a very specific hypothesis about the fact that when selfing arises, what is known as the effective population size of the population is halved, because individuals are now selfing rather than crossing between, and that has an effect on what is known as the genetically effective population size. It makes that population size quite small, and that makes populations very susceptible to what is called genetic drift, which for our purposes is just simply random processes can often take over, and selection is often very weak. and many genes that would normally be either deleterious or beneficial simply become neutral. And so what he's recently shown in a paper in Genetics is that the proportion of mutations that are arising across the genome that are neutral is significantly higher in the selfer compared with the outcrosser. And this is sort of consistent with the idea that island colonization and the transition to selfing is resulting in a lot more genetic drift in populations. Okay, I want to now just briefly talk about separate sexes, because this is sort of different now from selfing or hermaphroditism. I'll just point out that there are species in Ontario that have separate sexes in the wild. This is Thalictrum here, early meadow rue, which is very common in our woodlands throughout southern Ontario. And when it flowers in the spring, if you go out, you can distinguish males and females. Okay, so there's a female here and there's a male. And I got interested a few years ago in sex ratios. In other words, the prediction that goes back to the famous mathematical geneticist Ronald Fisher is that in an equilibrium population, a population that's been around and mating has occurred for a fairly long period of time, we should get 1-to-1 sex ratio. Okay? And many of you may be thinking, well, isn't that an inevitable consequence of the segregation of the sex chromosomes? Yeah, that is true to some extent, but that's the proximate mechanism that gives you 1-to-1. And of course, Fisher figured out why you have 1-to-1, and it is that is the sex ratio that maximizes the fitness of both males and females. So 1-to-1 is the optimal sex ratio for the population mean fitness. But the question is, what's the situation in plants? Do we find 1-to-1 sex ratios in wild populations? Well, a few years ago, we did a big analysis of as many data sets that we could lay our hands on. And what I'm plotting here is each one of these little circles is a species, and we're just plotting the mean sex ratio. Somebody has gone into a population, and they've just counted numbers of males and females and reported them. And this would be the equilibrium, 1-to-1. And what you can see is there's a lot of variation there. In fact, most species, 2/3 of the species, have biased sex ratios. And male bias is more common than female bias. Now, that's interesting, and of course it raises an interest. Why? I mean, why have we got these biased sex ratios in plants? Why aren't there more populations that are 1-to-1? Part of the explanation of that story of male bias is simply that males tend not to flower every year. Many species with separate sexes are long-lived clonal plants or trees, and when a female makes a lot of fruit, she wears herself out, speaking anthropomorphically. In other words, there's a cost to reproduction. And often what that female will do is take a rest for a few years and then flower again. Whereas pollen's cheap, relatively cheap. There's some phosphorus in there. But relative to making fruit, the cost of reproduction between the sexes is different. And so what we're seeing here is relatively trivial result. It's simply that most of these reports showing male bias is simply because males get to flowering earlier, they flower more often, and we also know that males don't suffer as much gender-based mortality. Females have a hard time when they're fruiting. It turns out, it's very interesting, that if you look at dioecious plants in general, there's a strong correlation with large, fleshy fruits, and they're very costly to make. Think of papaya, big papaya that you buy in the store. That's a dioecious species. And you think of many dioecious species have very costly fruits. So females don't tend to flower as often. So that becomes rather uninteresting, really. I mean, it's just simply, we're not really seeing the true sex ratio here. So what about this? Well, it's hard, a bit more difficult to argue that this is a cost problem. So we decided a few years ago to sort of look at the genus Rumex, the dock family. Up to now I've been talking about these amazingly showy insect-pollinated plants, and I thought it's sort of an irony that, as one moves into older age and, decrepitude, you find beauty in even ugly wind-pollinated plants. Sorry, David. He's a PhD student of mine who works on wind pollination. So, beauty is in the eyes of the beholder. Grasses can be very beautiful if you look at them carefully. Okay, so Rumex is this sort of pretty ugly plant. I mean, it's a wind-pollinated weed, but it has separate sexes. And so we've been putting a lot of effort into this species in recent years, looking at different species in different places, different postdocs and students. And all I'll say is that we've now kind of figured out what's going on, and that is that there is a bias that starts fairly early on. It's even in the pollen, and we use flow cytometry to sex the pollen, then at the seed stage, then at the seedling stage, and by the time they're flowering, the population has 90% of the plants in the population flowering are female. It's a strongly female biased sex ratio. So the question is, why? Well, we think it may be due to the sex chromosome system and the fact that in a system like this, the gametes, the male gametes, have an X or a Y, right? Because it's an XXXY system. And we know from work on humans, Drosophila, and many other animals, that the Y chromosome is degenerate. I don't know, those of you males in the audience may not know, but you've got a pretty crappy Y chromosome compared with the X. And there's a lot of interest in why Y chromosome degeneration occurs. We know it's largely to do with the fact that there are large areas of suppressed recombination, and bad genes just accumulate on the Y and are protected from what is called purifying selection. So what we've been very interested in doing is actually looking for signatures of deleterious genes associated with the Y chromosome as a possible mechanism causing this inexorable increase in female bias. And we've recently discovered in this group that there is in fact evidence. This was a paper that was published last year in PNAS. Okay, I'm going to finish very shortly. I know I've gone on a little bit, a few minutes too long, but let's go out with a bit of like naughty stuff, some sexotica, okay? And so I'm going to return here to natural history and tell you about just two stories that I guess, you know, are meant to wow you about plants, because I just think these are incredible stories. The first involves a species called taca, which I sort of got into through my visits to China. I go to China most years, and if you go to, you know, most people who go to China, you know, they go to Beijing or Shanghai, but I don't. I go to Yunnan and some of the really species-rich areas, particularly down near the borders with Laos and Vietnam and Thailand. There's some great rainforests down there, and in those rainforests, in those rainforests is this plant. And this has almost become a cult plant, by the way, in the States. I mean, the Americans, they love cults. And so among horticulturalists now, people love to grow taca, you know, to impress. As you walk in for a dinner party, there's this amazing plant on the table with all these dangly bits. And the question is, what is going on here? Now, if you're a creationist, it's easy. You just say, oh, God designed that. But that doesn't wash for an evolutionary biologist because, you know, if you take a Darwinian approach, you're going to say, well, wait a minute, it has a function. It's evolved by natural selection. And so we need to figure out what it's actually doing. And so we were, after this discovery, very interested in this, you know, enthralling, but curiouser and curiouser. This is a former student, Chris Eckert. Well, let me just tell you, we don't know the full answer here, but we've made some progress in studying this plant using genetic markers, and this is a collaboration with my two Chinese colleagues from Sichuan Botanical Garden in Yunnan. And so what we did was we sort of said, well, we need to kind of think about what the function could be. Maybe it's to attract pollinators. So the question then, you know, this thing is sort of shimmering around in a light breeze and it attracts pollinators. And that became a pretty good hypothesis when we discovered that the plant is being reported as being, and wait for it, sapromyophilous. And what does that mean? It means it's pollinated by carrion flies that are attracted to what they think is rotting meat. And apparently there's this convergent syndrome that has evolved in quite a few, where you get these shimmering structural parts that's supposed to look like maggots. And the idea is this attracts these flies along with smell, which is awful generally in these species. So we thought, well, okay, well, why don't we do a manipulative experiment? We'll cut them off. And you can do this without an animal certification scheme. You don't have to worry about waking up in the middle of the night feeling guilty that you've, you know, ripped out the sex organs or the parts of the plant That's why plants are so great for manipulative experiments. So what we did was a simple experiment where we compared the fitness, seed production and so on, of plants with and without these strange structures. It was a bust. Well, it wasn't a bust. Any result is interesting, but it didn't support our hypothesis. We got no difference between the two treatments. I then said, well, we better look and see if what the story is on the seed produced by these plants. And we found in these populations in China, we looked at 4 populations, that these plants are in fact highly self-fertilizing. In other words, if we use genetic markers to look at parentage, this isn't, at least it doesn't seem to be anything to do with attracting pollinators because they're mating with themselves. So we don't know what's going on here. So one issue is, of course, well, we are looking at the northern end of the range in China. Maybe we need to go to, further down into Thailand, into more southern areas. Maybe they function okay there. But right now, we have no idea what these structures do. And, generally in evolutionary biology, you don't invest in something like this if there's not a payoff. There's generally some trade-offs going on, and investing in these extravagant structures, these bracteoles, it can't be there, they can't be selectively neutral. That would be the hypothesis. So, last example. This one we did figure out, so we do have a solution here. So let me just, bird pollination 101, and this is, again, not being condescending, but some people are surprised by a factoid that I cover in this particular example. Turns out birds are very important pollinators. I think everybody knows that, and those of you who have bird feeders know that you have them because you like to attract hummingbirds. And if you're a birder, you know that hummingbirds are really important pollinators. But that's not a global phenomenon because there's no hummingbirds in Africa, India, or anywhere in the old world. They're restricted to the Americas. So if you go to Africa or you go to India and something is bird pollinated, it's not by hummingbirds. So we have a different set of birds in the old world that visit flowers. Hummingbirds hover, okay? But the birds in Africa and India and Australia, they perch. Okay, so they fly in and they land. Completely different foraging strategy. Now, with that in mind, in 2003, I was on a sabbatical at Cape Town. The project I'd gone to work with was a bust because the plant didn't flower. So I had to come up with another project. I don't want my chair saying, what did you do? Just spend the time drinking wine in wineries and hanging out? Oh, yeah, I got to publish papers from a sabbatical. That's what it's all about. So I went out to find a project. And I was very lucky to have a graduate student who was a great natural historian to go along with me. And one of the very first field trips, Bruce Anderson and I saw this plant. And it was, this very curious plant. It's endemic to the Cape. Believe it or not, it's in the iris family. So this is an iris. It's not in the genus Iris, it's in the genus Babiana. Babiana there's an adaptive radiation of Babiana in the Cape. There's about 70 species. A very small number of them are pollinated by, well, what? It's got red flowers, so that's okay. And so we were standing looking at this thing, and I said, well, what's going on here, Bruce? This looks like a very interesting plant. Why are the flowers on the ground, and what is this? Okay. So anyway, we said, well, if this is bird pollinated, bird pollinated plants generally don't put their flowers on the ground. That's a very risky place because of predation on the birds. So, very, very few examples of bird pollinated species with ground level flowering. But there are birds that walk around. I mean, maybe it's pollinated by a chicken or something, So highly unlikely, okay? So the question then became, well, let's sort of see if we can figure this out. So the question here is, what pollinates it, and what is this? Well, I'm sure most of you have thought this through, okay? It's a perch. And it's evolved to promote cross-fertilization in this genus Babiana. So it turns out that the only bird that visits this plant is the largest sunbird in South Africa, the Malachite sunbird. And what it does is it flies in, grabs the perch, turns upside down, and pollinates the flower. And so we were very excited when we took that photograph and we nailed down what was going. But you're not going to get a paper in Nature from just saying, hey, look, I found this interesting bird-pollinated species. You have to do an experiment. And so Bruce and I said, well, we need to do an experiment. And he said, well, what kind of experiment? I said, we cut it off, okay? And then we see how the plant does without the perch. And essentially what happens is you do get occasional visits to flowers, but it causes selfing, not cross-fertilization, because the bird is not optimally placed to bring in cross-fertilized pollen. And so by using genetic markers, we were able to show that this perch is a very efficient structure for promoting cross-pollination between plants and maximizing fertility. So anyway, that work was done by Bruce Anderson and also by a master's student in my lab. And it's a remarkable example of the evolution of a very specialized pollination mechanism that actually involves not a part of the flower so much, but what was the structure of the inflorescence. And through the course of evolution, that inflorescence, the sister group, has flowers all the way up the perch. And those flowers have been lost through the course of time through mutations, and we now have a single perch. Okay, so what are the take-home messages? I hope I've shown you that natural history observations are still alive and well despite the genomics era. And, you know, we can actually still do classical Darwinian natural history and make discoveries and publish those in the top peer-reviewed journals in the world. It's just a question of going out and discovering things and looking carefully. However, I also want to make the point that I think field experiments and genomics, when combined with natural history, provide a very, very powerful approach to sort of decomposing problems of adaptation in plant reproduction. And then finally, I'll just say that plant sex never stops. You know, it goes on all the time, like evolution never stops. I'm often at a cocktail party and somebody will come up to me and say, what do you do? And I say, I'm a teacher at U of T. And they say, oh, what's your area? And I say, I'm an evolutionary biologist. And usually the first thing that comes out of their mouth is, oh, do you work with dinosaurs? You know, I mean, it's sort of like the public's perception of evolutionary biology is that you have to work on bones. But what I want you to understand is that the majority of evolutionary biologists are studying evolution now. Evolution is occurring in your stomachs right now because you've got more bacterial cells in your stomach than you have cells in your body, and they're evolving. In fact, the best place to study evolution is just walk across the road and go to the hospitals. There's all sorts of interesting problems there to do with antibiotic resistance and so on. So evolution never stops and plant sex never stops. And if you go out next spring and you have a look at flowers, you could become a plant sex voyeur like me. You know, it's fun to go out there and have a look at those bits on those flowers. And you never know, it might change your life. Thanks very much.